Hello everyone, I'm Julong Wei and a postdoc from local Region Wayne State University. Today I will present our current work, single-cell multiomic analysis of the gene regulation across context dissects the molecular underpinnings of GLBIO in inflammatory diseases. And the GLBIO interaction between played an important role in inflammatory diseases. Evidence has shown that the mismatch between the genotype and the environment would lead to high disease risk, especially in today's alteration of lifestyle and diet and drinking coffee and smoking. In our previous study, through the in vitro system with 15 treatment in 5 cell types, we discovered 215 genes with GLBIO interaction over 40% of associated with diseases in the GWAS. One hypothesis is that the genetic variants contribute to the diseases by perturbing the transcription of the gene expression while altering the transferrin binding to the regulatory elements to causing the misregulation of the gene in a cell type specific or environment specific way. And over 19% of GWAS are in non-coding regions such as enhancer and promoters, profiling the chromatin accessibility using a technique were able to capture these genome-wide cis-regulatory regions. Most recently, several studies, such as in the recent paper using a single-cell multiomic data, revealed that the causal variants for the gene expression and disease risk are enriched in these regulatory genomes. Integration of this knowledge would help us fine map the causal variants and better interpret the molecular mechanisms. And most studies focused on the cell type specific regulatory network using the single cell experiment. Yet few studies explored the context specific regulation patterns at the cell type resolution. This will limit our understanding of the regulatory variants contributing to the diseases in a specific environment. And to resolve this question, we use the PBMCs from the 12 participants activated with phytohemoglutamine and treated with respective compounds, caffeine, nicotine, and vitamin A, D, E, and zinc, and also treated a pair of control treatments, ethanol and water. Using the single-cell multiomic kits, we generated a total of over 60,000 high-quality cells for the INC and TechSeq with the detection 32,000 genes and over 260,000 peaks, and were measured from the same cells. And through the routine analysis, we obtained the high resolution of cell type annotation for 11 subclusters. With the combinations of the gene expression and the chromatin accessibilities, the cells in this largest clusters were well positioned into the 6 subtype of the of T cells. CD4 and CD8 and the central memory and the effector memory and the double negative and the mucosal associated T cells. This inspired us to ask how the chemical compounds shape the transcriptional accessible chromatins in different immune cell types, and what transfactors are involved in the gene expression changes in response to exterior environmental stimuli, and how the genetic variants interacted with the environment influence the gene expression and diseases. And first, we want to know how these compounds change the gene expression and chromatin accessibility in different immune cell types. And to answer this question, we performed the differential gene expression analysis to characterize the gene expression changes in response to stimuli in immune cell types by the dissect tool. And we identified a total of 6,500 unique differential expressed genes deaths across cell types and treatment. And the caffeine induced the most immune response, over 4,700 days, followed by the nicotine with 33 unique days. We also observed the cell type specific gene expression changes, such as we detected the most days in the monocytes, a total of over 3,300 days. And next, we want to know what the biological processes are involved in the transcriptional response. We performed the gene ontology enrichment analysis of this differential expression genes across cell types and treatment for the upregulated genes and downregulated genes separately. For the upregulated genes, we observed the the metal iron related pathways are enriched in all the immune cell types treated with the zinc, such as cellular response to zinc or iron pathways, as expected. This pathway included mesolusal iron family genes, which play important roles in regulation of the homeostasis and the detoxification of level of the heavy metals in the individual cells. And the downregulated genes are enriched in immune regulated pathways, which implicated the widespread roles in immune system for these factors. And next, we investigated how the chromatin accessibility changes were induced by this exterior treatment in immune cell types using a differential accessibility analysis. In total, we identified over 70,000 unique differential accessible regions DAS across cell types and treatments. We observed a greater number of the DAS in caffeine and nicotine in most cell types, and other treatments have a smaller number, a smaller discovery DAS in most cell types, and except the vitamin D in the monocyte. Among other conditions, we identified the most number of the DAS in the vitamin D in the monocytes, over 100 DAS. Monocytes also show the most response changes in chromatin accessibility, with the largest discovery of DAS over 7,200 DAS. And next, we perform the enrichment analysis to examine whether DAS are enriched in whether DAS are enriched in DAS in the corresponding conditions through the Fisher exams. We annotated peaks to the closed genes, and for each condition, we count how many genes have DAS and are not for differential expression genes or background genes. The forest plot in the right panel visualizes the log odds ratio with 95% confidence interval. We can see the DAS are significantly enriched in DAS in most conditions. We identified the 24 conditions with significant enrichment. Now we take a closer look at the example condition monocytes with the zinc treatment. In the condition of the monocytes with zinc, the gene expression response changes significantly correlated with those of the chromatin accessibility, with the Spearman correlation as the coefficient is 0.09. Here, the red color representing the significance only the chromatin, the blue is significantly only in the genes, and green representing the significance in both the chromatins and the genes. We also annotated all the mesolusal iron family genes in the scanned plot. Then we visualize the gene track plots for this mesolusal iron family genes, show the significantly higher expression values in the gene expression and the chromatin accessibility in the zinc treatment. So this result suggests the chromatin accessibility in the cis regulatory elements of the genes are the main driver of the regulation of the gene expression. So we want to know what the transfactors are involved in gene regulation processes in immune cell types. To do this, we first quantify each transfactor activities for each cell. We measure which measure the genome-wide chromatin accessibilities with a particular transfactor motifs binding sites. For each combination, the cell type and the treatment and the individual, we calculated the average motif activity across cells from the same combinations. Next, we perform the differential motif analysis using the linear regression model and identified the 294 response motifs. And then we selected 111 response motifs with the most activity changes to explore the mechanisms underlying the changes in the TF activities. In the heat map, we can see 6 cluster patterns for these motifs. The motifs in the class 1 have the largest correlation between the TF gene expression and the TF activity. This cluster include the TF transfounded in the canonical type 1 interferon signal pathways. And given the above analysis, we assume that this response transfactor motifs may be the potential target elements of the GLBIO interaction. So by leveraging this response motifs, we try to explore the molecular underpinnings of the GLBIO influencing the gene expression and the diseases. At the genetic regulation of the gene expression level, we want to know which latent variables can be captured in the EQTL cohort with the unknown or uncharacterized environmental factors. To address this question, we annotated the genetic variants using our single-cell multiomic data. The regulatory variants, which in the cell type activity peaks, were partitioned into the 29 categories of response motif annotation, representing the different potential latent environmental variables. After that, we performed the enrichment analysis for the EQTLs from the GTEx, the whole blood tissues using a torus. The forest plot in the right panel shows the log odds ratio with 95 confidence interval. We identified the 22 category of response motif with significantly enrichment for the EQTL signals, especially in caffeine and nicotine. The top 5 enriched category of the annotation includes the monocytes with vitamin D, and the NK cell with nicotine, and CD4 with caffeine and nicotine, and the effector memory T cells with caffeine. This result, the potential latent environmental variables exist in the EQTL datasets. And our data also offer a unique opportunity to explore the molecular mechanism behind the risk genes. To do this, we performed the TWAS and correlation analysis for the 11 inflammatory diseases using a 5-map EQTLs with the integration of this context-aware annotation. We use the intact a model-based approach to combine the evidence from the correlations and TWAS analysis to identify the putative causal genes. At 5% the FDR level, we identified a total of over 860 GWAS loci and across diseases, and for 43% of these loci, the causal range was annotated in the response motifs. And next, we investigated which the category of the response motifs have more risk genes than expected. The heat map shows the log odds ratio observed was the expected value across conditions and diseases. And we can see the nicotine and in the NK cell and vitamin D in the monocytes and zinc in the double negative cells have more risk genes than expected for the most diseases. We also find that some categories of annotation shows the trait-specific pattern, like the annotation of the NK cell with the caffeine has the enrichment in the rheumatoid arthritis, and the T effector memory cells with nicotine only enriched in the coin diseases, and the CD4 nail with zinc showing enrichment for the systematic lupus erythematosus and rheumatoid arthritis, and double negative cells with caffeine are enriched in the coin diseases. So this enriched category of notation suggests the more important roles of the latent environmental factors in etiology of the diseases. So lastly, in this study, we conclude single-cell multiomic captured the cell type specific patterns for the transcriptional and chromatin response changes to the environmental factors. We demonstrated the transfactor gene expression is one of the mechanisms driving the transfactor activity changes. And using the genetic variance annotation in the response motifs, we identified the latent variables for the EQTL in the data with unknown states. And the integration of the context-aware annotation offers a unique perspective to study the mechanism of GLBIO in contributing to the diseases. And to the end, I want to acknowledge all the members of Luca Pico-Ridge Labs, and especially my PIs, Francisca Luca and Roger Pico-Ridge. I also thank Mohammed. He has done a lot of work for this project. So we truly appreciate all your patient listening. Welcome your question.
Quick question. Just wondering if you look at DNA methylation and negative modification, and whether or what kind of challenge you would have using
So could you repeat?
DNA methylation and negative modification.
No, in this project, we just look into the performing accessibility. We don't investigate the DNA methylation or other histone correlation. Yeah. Thank you. That's a good question. Yeah.
So in one of the earlier slides, you asked whether the DARs are enriched in the DEGs.
DEG in the DAR.
Yeah.
So it's
for the same condition.
Yeah, the same
Did
condition.
you look at for mismatched conditions? Are there things that just change regardless of the condition?
Oh, that's a good question. But it's the same condition. We just compare the same condition. Yeah. Maybe we go back, we can look into the correlation between the in mismatched condition. Yeah. Thank you.
Just a really quick question. I was curious, when you're looking at the SNPs to compare to the ETL study, was that they used genomic sequencing on the samples that you had collected, or were those?
I use the different cohort. We just use our data to annotate the genetic variants. Then we use the genetic variants annotation, then to use the GTEx, GTEx whole blood tissue equity cohort to test if enriched.